I'll begin by introducing you to the Plant Phenome Journal webinar series. I think this is our 14th. I've sort of lost count at this point. Today we are, I'm very pleased to say we're joined by Dr. Ian Stavness. He gave some excellent talks at Phenome meeting, but he was also a finalist for the North American Plant Phenotyping Network Early Career Award, which is how I first saw his work and was really impressed. So Dr. Stavness got his engineering degree in electrical engineering and a BS in computer science from the University of Saskatchewan. He got a master's in computer engineering from University of British Columbia, a PhD in computer engineering from University of British Columbia, and I love his dissertation title, Bite Your Tongue, B-Y-T-E. He then did a postdoc at Stanford in bioengineering, and since then he's been at the University of Saskatchewan, and he's now an associate professor. So he's going to be talking to us today about deep learning for image-based plant phenomics. I'll encourage everybody to mute your microphone. I will be muting those who I don't see muted. You should be muted on entry. But other than that, Dr. Stavness, please take it away.
Great. Thanks very much, Seth. It's a great opportunity to speak at the webinar series here today. So in this general area of image-based plant phenotyping, we sort of categorize the types of phenotypes you might want to extract from images into a few categories. So some of the let's say, easier ones to do, we would say, are linear functions of plant pixels, or nonlinear functions of plant pixels, but are sort of geometric statistics. So maybe the convex hull of the pixels or the center mass of the pixels. So these things on the left side here, we categorize into what we would call, could be done with what we say is traditional image processing pipelines. So there's great software to do this, such as PlantCV, where you can do a series of image analysis steps to get to your result. So things like maybe color correction or histogram normalization, thresholding, these sort of steps in the process that give you a result that's quite reliable and for these sort of what we call simpler phenotypes. But there is a class of phenotypes that are much more difficult to extract from images, and these are nonlinear functions of plant pixels and also non-geometric descriptions. So all of these things on the right side here, so counting leaves or organs of a plant, trying to assess the response to stress, the age, the growth rate, or even more downstream phenotypes like biomass or yield. And this category of phenotypes, we really think that deep learning approaches have a promising approach in this area. So for more difficult ones, we think these deep learning approaches, which really have taken over a lot of computer vision research these days, can be quite useful. The other area where we see the distinction between sort of traditional image analysis and deep learning is indoor versus outdoor imaging. So in indoor imaging, things are fairly controlled. So again, we think that image processing pipelines make a lot of sense there. But for outdoor imaging, the images are quite a bit more variable or even chaotic, you can imagine. And here we also see deep learning having a promising approach. So in particular, in outdoor imaging, you have much more variation in terms of lighting conditions. You have plant motion due to wind, sensor motion, because usually the camera is either flying or driving over top of the field. As well as plants that are very close together, so lots of problems due to occlusion or cluttered backgrounds, those sort of issues. These deep learning approaches we think are much more well suited. So in the talk today, I'm going to talk a bit about a piece of software we've been developing called Deep Plant Phenomics. So this is a platform that we hope is going to make it easier for plant phenotyping researchers and plant scientists to access some of these deep learning approaches. So it's an open source software project, and you'll see a number of the techniques that I'll talk about today. They're sort of available to be used through DPP, as well as some of the data sets or loaders for some of the public data sets that are available. So in the talk today, I'll talk about our work in leaf counting, sort of our initial work using deep learning in phenomics. Some of the work in outdoor imaging, since that was the main motivation for us to pursue these areas of analysis, and then some of our initial projects at analyzing those outdoor pictures. So starting with leaf counting, as a sort of general primer on these deep learning approaches, sort of the workhorse or the main approach that most people are using when they say they're using deep learning are something called convolutional neural networks. So these are neural networks that have a particular structure. And you can see in this example cartoon here, the idea of convolutional networks is that you have filters that are convolved or sort of passed over the image to extract features, or we'll call activation maps from those images. And this sort of process of convolving an image with a set of features to get activation maps is done repeatedly in a convolutional neural network. So this particular block, this little process, is repeated many times. So at the start, it's done on the original image, the RGB image or other bands that you have. These convolutional layers are applied to it, and then there's downsampling layers, here called max pooling layers, that are basically downsampling the image, and then you're running another set of convolutional filters over those downsampled images, and then repeated. So downsample again, convolutional blocks, downsample, convolutional blocks. And at the very end here, you might have some more traditional neural networks called fully connected networks to make some estimation of what you're trying to get out of the picture. The main advantage, or the main reason that these CNNs work, is that they're able to capture local features in an image at multiple scales. So that's why the downsampling occurs. And the main difference between traditional image analysis pipelines and these sort of convolutional neural network approaches is that in traditional image analysis, you have to choose particular parameters and sort of features that you want to extract from the image. Whereas in the deep learning approach with CNNs, you provide example images and you train the network to learn the features that are most appropriate to extract for the task at hand. So we call these learned features, or the filters have learned features in them, and this is really what makes CNNs more appropriate for these highly variable and very difficult computer vision tasks. So our first foray into applying CNNs to phenotyping was in rosettes. So this was done using the International Plant Phenotyping Network data set for rosette images. The kind of one of the really nice aspects of IPPN is the ability for them to propose these data sets, and allows many people to work on different techniques for doing phenotyping tasks. So certainly a lot of the work is made possible by IPPN and other public data sets that are there. In this initial work, we had three main things we tried to estimate from these rosette images of either Arabidopsis plants or tobacco plants. So counting leaves, also estimating the age of the plants, so hours after germination. And then for the Arabidopsis data, there's 5 different mutant classes, so also trying to classify those mutants based on the pictures. So this work was published in our DPP paper, and we were able to show very good leaf counting, so low error rates on leaf counting from these rosette pictures, a reasonable age estimation, and then very high accuracy for mutant classification. So this was really just kind of an initial demonstration that yes, these CNN approaches can be used in the context of plant phenotyping, and within the context of very small data sets, because the IPPN data set still has a very small number of pictures of rosettes. Based on that initial work, we submitted a entry into a leaf counting competition. So this competition is run also by the IPPN, and this was at the CVPPP workshop in 2017. So CVPPP is Computer Vision Problems in Plant Phenotyping workshop. And I'll just make a plug that they're having the 2019 version of this workshop at CVPR this year. So I would really encourage everyone to submit work in this area of image-based phenotyping to CVPPP, because it's a great workshop and it's a good chance to particularly collaborate between the North American researchers and the European researchers, because the IPPN group is very strong in Europe. So for the competition, this was done again with a sort of smaller data set of the rosettes called the competition data set, and we used a very similar approach, but this time we also did an initial step of segmenting the leaves from the background of the plants. So we had one network to do the binary segmentation of plant versus background, and then a similar CNN to estimate leaf counting, as was done in the previous work. So the motivation for doing the background segmentation was because for this particular data set, there's 4 different subdirectories of images, and they all have very different variation in their leaf counts. So you can see that each subdirectory has a different sort of range of leaf counts, different distribution, and they have very distinct backgrounds as well. So the A1 directory has these orange trays, for example. So we didn't want the network to sort of pick up, try to learn which subdirectory these images were from, and then base the count on that. We really wanted to try to focus on the plant themselves, try to focus on those for counting. So that's why we did the background subtraction in this work. So this background subtraction or segmentation network uses this encoder and decoder style architecture. This is very common for segmentation networks. And this was based on SegNet. There's other architectures. UNet is very popular as well, but all have this general encoder approach and then sort of a mirrored decoder that takes you from the sort of internal features up to probabilities for each class in the output image. The nice thing about SegNet or some of these fully convolutional networks is that there's no fully connected layers, and those fully connected layers have the majority of the parameters. There's many fewer parameters when you just use convolutional neural networks in the entire architecture. And so that allows us to train from scratch on small numbers of pictures. And the results for segmenting plants from these data sets were quite good in all of the Arabidopsis directories. The A3 directory had quite poor performance for segmentation, and these were the tobacco plant images. And you can see an example here in the middle where the tobacco plant segmentations are quite poor. So some of the darker regions on the tobacco leaves are not segmented correctly. So it could be the tobacco plants have quite different structure, so the network wasn't able to generalize to those as well. And also there's the fewest number of images in that A3 directory, so it could be just the lack of training data for those plants. And then the counting by regression is very similar CNN that we used before, except here now, in addition to the RGB image coming in, we have the S, or the segmentation channel as well. So there's 4 channels coming in, and then we try to estimate the single leaf count at the output of the network. This is similar to VGG16 architecture. We did some padding tricks to reduce the parameters, and we also trained this from scratch on the IPPN data set. And in general, we had better results than the previous competition, except for that A3 directory with the tobacco images. And in fact, we placed second place in this competition with these results. So in general, again, it showed the strength of CNN approaches for doing these more difficult phenotyping tasks. But we did a kind of a lingering question. We went to a lot of effort to do these segmentation in addition to the counts. We really wanted to know if it was really important to do that or not. So was that segmentation channel actually useful? So to pursue that question, we did an ablation study. So we tested head-to-head just using RGB images into the counting network versus using the RGB plus the segmentation channel. And the main results we found was that in that A3 directory where we had the worst counting performance, if you didn't use a segmentation channel, then the performance improved substantially. In all the other subdirectories, there wasn't a substantial difference between using the segmentation channel or not. But in that A3 directory, it was a big benefit. And again, this was the directory where we had very poor segmentation masks from the segmentation network. So the sort of take-home message from this work, we feel that really doing the segmentation for this task didn't really improve the results very much. But if you had poor segmentations, it could reduce the accuracy quite a bit. So you should be definitely careful to make sure that if you're going to use a segmentation channel, that it's quite accurate. Another general challenge with this leaf counting problem with IPPN data, but generally that we face in plant phenotyping, is that the data sets we have are still quite small. So in general computer vision tasks, usually the data sets are in the millions of images, whereas we're usually working in the hundreds or thousands of images for plant phenotyping. And that's certainly true in the IPPN data set, where we have hundreds of images in total in the data set. One way to improve or try to get more data is through data augmentation. So there's simple augmentation approaches where you take the input images and do things like change their brightness or contrast, flip them or rotate them, or do other sort of transformations to kind of give a bit more variation to the images to help kind of get more training data so that the network is sort of more generalized when it's trained. It doesn't just fit directly onto the small set of images and overfit them or memorize them. We use those sort of augmentation approaches, but we've also explored using computer models to help with augmentation as well. So in this work, we generated synthetic or computer models of Arabidopsis rosettes and then used the pictures of those as extra training data. And the advantage of using models is that you can now sort of directly generate models that have particular properties, so you can kind of fill in the gaps where you don't have example images, and should help the networks to generalize to those examples. So this was in collaboration with a group at the University of Calgary, and the models were L-system models that were used to create these Arabidopsis pictures. So here's some examples of real rosettes from the data set at the top, and then the synthetic ones. And so certainly we can create with these rosette models data that has sort of the right sort of structure, so the right placement of leaves, or sort of physiologically correct placement of leaves and size of pedicels and such. And for this particular task, the appearance of the models is not so critical. The images themselves are quite low res, so there's not a lot of texture in the leaves. But this is certainly one challenge with models, is that you can add a lot of variance and a lot of examples through synthetic data, but it's important that the synthetic data is sort of sufficiently representative that it's useful for training. For this task and these models, we found it was useful. So if we trained with just synthetic images and tested on the real rosettes, we had good results, and vice versa as well. So if we trained just on the real image and then tested on synthetic rosettes, we were able to count leaves quite accurately. And then the best results we found was whereas if we mixed together real and synthetic rosettes, so the most training data we could get, testing on real rosettes, we had the highest correlations. So we do think these model-based augmentations are useful, particularly in cases where you either have a problem where the structure of the plant is most important, or you have models that have very good, very faithful representation of the appearance of the plant. So those are a few projects in leaf counting, but as I said, we were really motivated by outdoor imaging as the main place where we think we'll have a lot of benefit of using these sort of deep learning approaches. So I'd like to talk a little bit about some of the outdoor imaging activity here, and then I'll talk about some of the analysis projects. So as was discussed with Seth a little bit right before we started, the work here at the University of Saskatchewan in plant phenotyping is being undertaken within this Plant Phenotyping and Imaging Research Center, we call PERC for short. And this has been quite a nice opportunity. We've had a large grant from the Canadian Science and Engineering Research Council to bring together a research group across agriculture, engineering, computer science, and social science to develop expertise in this area of image-based plant phenotyping. It's a 7-year project. We're finished the 3rd year, so we're just transitioning from the first phase to the second phase. So this is a time where we're still very much open to collaboration with external groups. So we do hope that if you have an interest, you can contact me, and we have lots of opportunity over the second phase of the grant. But really the target here is to develop tools for image-based phenotyping, really specifically targeted towards plant breeding. And the connection to plant breeding comes because we have strength at the University of Saskatchewan in large crop breeding programs. So these are images showing field trials from Curtis Bosniak's wheat breeding program. And then we also have a strong connection into pulse breeding programs with Kirsten Bett, and canola breeding at Agriculture Canada with Sally Vale. So since we have this resource of outdoor field trials, this is where we're sort of focusing on tools to try to get phenotypic information that's useful for plant breeders from these contexts. The majority of images we have are aerial images. So Steve Shirtliff here is sort of leading the group on aerial image acquisition, primarily with drone images. And using drones, of course, we can get nice pictures, sort of standard approaches to get pictures of an entire field, an orthomosaic sort of stitched together from the drone pictures. But for plant breeding and other agricultural science, usually we need to do analysis on a level of a subregion within a field. So, for example, within a plot. So, for a plant breeding trial. So you can see these black boxes are really what we need to get to be able to analyze inside of those images or even subregions within the plot to get phenotypic information that's actually useful for plant breeding. So phenotypic information on a particular variety or a particular treatment in the field trial. So we've been developing some software that helps us generate those per plot images. So we call this software Plot Vision, and it's a web-based tool, so it allows you to upload drone pictures sort of as painlessly as possible. So it's a way to get images up into cloud storage. In the background, it can then process images and do automatic orthomosaic generation using Agisoft Metashape. So that'll kind of happen. You connect to Agisoft in the background and then generate orthomosaics that you can view. But then there is helpful tools to generate these per plot bounding boxes. So given the layout of the field, in this particular application, you just click on the corners of the field and then it'll sort of overlay the rough outline of all the plots. And then from there you can manually move things around if you want to sort of adjust things slightly, or maybe erode the plot, so you're just looking at the inner region of each plot or a subregion. But there's sort of a sequence of tools to help us go from drone pictures right to the individual images for each research plot. We've been doing other types of image acquisition as well. So one approach we call camera on a stick. So we have time-lapse cameras that capture pictures continuously through the season. So this is some images from an early commercial security camera that we had out in fields, and these were deployed on a diversity panel for lentils in Saskatoon, and as well as in Italy and Nepal to get to look at very different growing seasons, really different day length over the season using these high temporal resolution pictures. And then with our engineering colleagues, we've been developing sort of a cheaper and custom solution of camera on the stick with a Raspberry Pi that can be deployed into the fields here with a solar panel and battery, so sort of self-contained camera solution. For ground-based imaging, there are groups here in PERC that are developing more fully fledged sort of phenotype mobiles or phenomobiles, if you will. In our group, we've been trying to deploy very cheap and easy imaging systems. So really we're putting action cameras on sticks and either walking them through the field in the case of a GoPro, or sticking them on the back of sprayer booms for a tractor, which we call the protractor. So the advantage of these kind of what we would call sort of quick and dirty imaging approaches is that, there's all the 6 cameras on the back of the tractor there. So these are very inexpensive imaging systems, and they're very easy to operate. So even aerial drones require, in various jurisdictions, different amount of certification or training to fly, whereas you can send literally anyone out to the field with a stick to walk through it, or very minimal training on a tractor to drive through. And they're also very easy to replicate. So since they're inexpensive and just use sort of standard consumer version cameras, you can replicate them at many sites. So if you have 10 different trial sites, instead of moving your phenotyping equipment between trials, you can just replicate them very easily and have one per trial or one per location. The difficulty with these quick and dirty approaches is that they're quite dirty, so the data you get is not easy to use right away. So the GPS is very inaccurate. We don't have a real-time GPS on these. We just take the GPS off the camera, so we have quite inaccurate GPS. There's much more camera motion, particularly in the handheld version. As the person's walking, they're going to have more camera motion than on a more dedicated phenomobile. And if you have multiple cameras, they're not synchronized. I know we just kind of let them take pictures. And we see here a trade-off between sort of the complexity of the acquisition system versus the analysis system. And since we're computer scientists, we think that it's easy for us to try to move some of that complexity away from acquisition into analysis. So we're more interested in getting a larger set of images that might take us a bit more effort to try to analyze. So here's some videos showing this is the GoPro in action. So a computer science summer student sent out to the field to get some exercise and enjoy the nice weather in Saskatchewan in the summer, walking with the GoPro here. So it's really just a GoPro camera on a gimbal. So it's stabilized a little bit as walking, so it sort of should point downward most of the time. And this is a video from the camera of the set of time-lapse photos taken. This is for lentil plants. And these are images, so after taking all of those images and using the similar sort of orthomosaic generation, these are the types of pictures you can get for early season wheat. And this is early season canola. But certainly one thing we found is that the stitching process is quite challenging. So generating the orthomosaics with drones, you have sort of a few pictures over the whole field. Whereas if you have very close-up images now, even just over a section of the field, like a plot or a range in this case of rows, you have many, many images to stitch. So this is certainly a bottleneck that we have to face, as a challenge. So that's why we've moved towards a tractor system where we can take pictures sort of simultaneously across and cover at least the whole width of the rows. So this is an example of the pictures together. So they're not precisely synchronized, but they're sort of approximately synchronized. That would help us sort of get the full view of the row. So we can sort of stitch horizontally quite easily because the pictures are taken quite close together. But then we still have the problem of we'll see those same rows multiple times. So if this is the row of interest, as the tractor drives, each picture you'll keep seeing that row again and again. So we want to be able to detect unique rows so we can get all of the pictures of those rows. So we can do this very straightforward manner with kind of an image processing pipeline where we first pick all the green pixels and find their peaks to get an approximate location of the rows. From there, we can fit the center line for each row. And then between sequential pairs of images, we can do sort of a co-registration and find the overlapping rows here. So you can see when a red box pops up, that's a new row that we haven't seen before. And then the purple ones are overlapped between the previous picture. And from that, we can then pick out all of the rows that belong to all of the images that belong to the same row. So there's 4 sets from this row as the camera drove over. This is a different row, and this is a different row. So they look very similar, but they're actually at slightly different camera angles because they'll be taken as the tractor moves across the row. But with this fairly simple imaging pipeline, we can get per row images, which we think are useful for, at least for early season emergence, early vigor. This particular approach does right now rely on the fact that you have good distinction between rows. But we think that there's ways to extend this to later season images as well once you have canopy closure. And then the other aspect of some of the outdoor imaging tools we're developing are visualization tools. So certainly most of the plant breeders they certainly want numeric data, so they want phenotypes in a spreadsheet that they can do analysis on. But we think there's also value in storing images to be inspected later on. So after the growing season, plant scientists can come back and maybe verify the notes they've taken from the season, or just go back and double check, if they find some unique line from their analysis, they can come back and check out the pictures of that line over the season just to verify that they're seeing what came out of the data analysis. So this is an example application. Again, it's a web-based tool, so you can go to this website, pick a particular trial you're looking at, and then in this case, these are single row plots. So this visualization along the top gives you a view of the whole range, so all of the rows, and then the second picture down is an image of the entire row, and then the bottom image is a zoomed in portion of that row. So it gives you a visualization that provides you sort of very detailed pictures of the plants in the context of the overall row, and then that row in the context of the overall set of rows or ranges. And then you could pull the slider on the left to go through dates. So you can start in early season, and then you can see the next time point, the next one, all the way up to flowering in this case. So we think there's value both in the analytic tools, but also in these visualizations just of the images themselves. So both for the ground-based imaging, also for the time-lapse cameras, we have sort of custom tools to allow you to inspect those time-lapse pictures more readily. Great, and so I'll finally talk about some of the analysis we've done for these outdoor images. So the main thing we've focused on so far is counting plants or plant organs like flowers, etc. In this work, we were looking at doing early season wheat emergence counting, and we chose this because we thought it was probably the most difficult emergence estimation because of the thin overlapping structure from the wheat leaves. So we were using, for imaging data here, these high-resolution GrowPro images. So we got quite high resolution of each individual plant. And then the labels that we were looking at, we had plant scientists look at sort of small patches within the image and estimate how many plants they could see from the picture itself. So the labels were for each small region of the image, a scalar number, which was the count, the number of plants. So we think this is quite a difficult challenge, especially if you compare that to leaf counting. So this is an example from the IPPN data set where you're counting leaves. So the labels are very similar. It's still a scalar count for the number of leaves in that picture. But it seems to us that counting the leaves is probably a slightly easier task than trying to count plants from this sort of tangle of wheat plants. So just for intuition, to see how the plant scientists were counting, we asked them to sort of give us some clues as to what they were doing. And so the first thing they said, they looked for at the base, they tried to count the base of each plant, so as many stems as they could find very close to the ground. And then they estimated as well regions where there was sort of dense plant matter. They thought there's probably additional plants underneath those that are providing leaves, more leaves given this growth stage. So they gave us this intuition for a few images sort of annotating what they were looking at. But the real data that they gave us was just the count per patch. And we used sort of similar deep learning architectures as we did for leaf counting for this data set. From this very high resolution image of the whole plot, we used a segmentation network to try to roughly segment the leaves from background. And then based on these segmented masks, we could then identify patches of images. So that's why these patches are sort of connected areas where we see leaves, and those patches are the red boxes here. Those are the patches that were counted by the plant scientist as training data. And then, so that was used to train this counting network, so a similar counting by regression approach as we did for leaf counting. And so then per patch, we estimate a count and then sum those counts up to get the final count for the overall plot. So there are sort of two aspects of the network: for the first initial segmentation, and then the secondary plant counting from each patch. So for the segmentation results, the accuracy was sort of reasonable. Again, it's a difficult segmentation task because of the sort of thin leaves that are overlapping. And in this case, we were really interested in sort of a rough segmentation. So we wanted to make sure that we didn't cut out any plant material, but if we had a little extra soil, it didn't really matter too much in this case, because we were mostly using it to find patches that we could pass down the network, smaller patches. And then on the plant counting, we achieved quite reasonable accuracy. Again, this is sort of the first baseline for this type of data set. But if we look at it compared to the leaf counting results in sort of normalized accuracy, we were slightly better than what we were getting for counting leaves from the rosette images. So we thought this was did a reasonably job of doing plant counting for these early season wheat images. Of course, from here we want we need to test this approach more widely on different data sets. So we consider this sort of a baseline, and the data for this and our software are available. So we're hoping other people can kind of use this data as a benchmark to try out different approaches for counting plants. One of the challenges with these sort of deep networks is that they learn the features that sort of are best for the task at hand, but it's difficult. It's not necessarily true that the features that are learned are going to be very intuitive. So we might not know what the network's actually picking up on in the image to accomplish the task. So this generally can be a bit it's challenging because it's hard to have a lot of trust necessarily in these networks. We don't really know what they're doing. So one of the approaches you can do to provide a bit more intuition is to use visualization approaches with these networks. So we did this. We used a saliency map visualization for these plant counting images. So these sort of extract a heat map from the low-level feature maps to say which regions of the image are kind of is the network sort of focusing on when it's accomplishing the task. So in this case, what are the regions of the images that the network's focusing on when it's trying to make a count of how many plants are in there. So you can see them superimposed here on the right side. And what we found was that the network seemed to focus on the base region. So in the top example, the base of the plants is on the top edge of the image. On the bottom example, the base is on the bottom. So the heat map regions are towards the base of the plant. So that sort of aligned well with what the plant scientists were doing. And then there are regions of sort of overlapping leaves you can see that are also focused on, and that also sort of mapped to the other aspect that the plant scientists were focusing on when they were counting. So these visualizations can help you sort of ensure that the network seems to be looking at the right part of the images, and can help with providing a bit of trust as to how the network is working. And we've done similar things, counting methods with wheat spikes from close-up pictures. And again, the saliency maps here show that the networks do focus on the wheat heads themselves. And this helps you show that at least that the network's not just memorizing the training images and sort of overfitting on them. And it gives you some hope that it's focusing on unique things in the images that will help it generalize to other images that it hasn't seen before. And then the last project I was going to talk about was trying to make these networks more efficient. So as I said, that one of the challenges we have is usually small numbers of images. And so then the tendency is with a very complex network that has many, many parameters that it can train, so it can kind of memorize those examples that it's seen and then do very well on those examples, but then very poor on a novel testing image that it hasn't seen before. And one approach to try to reduce that overfitting problem is to try to make the network smaller or reduce the number of parameters. So in particular for these sort of segmentation networks, you have an encoder part and a decoder part, so you really have sort of twice the computation and twice the number of parameters as you would just with the encoder part if you're training end-to-end. So in this approach, we replaced that decoder part with really simple operation to go from the low-level features back to the segmentation mask. In this case, it's a depth-to-space operation. So you sort of take the filters at the lowest level that have lots of depth, you have many filters, and rearrange them spatially to get up to an image. And this rearrangement operation sort of replaces the whole decoder and provides basically half the size of the network approximately. So we tested this on satellite images for a road extraction challenge in last year's CVPR and found that these sort of depth to space operations got very similar results to using the full segnet for these sort of operations. So we think that doing the similar thing for vegetation segmentation, such as trying to segment all the plant material from a large orthomosaic of a field, also can be done with these simpler networks, because the segmentation problem itself is quite simple. It's just a binary segmentation of plant versus background. We have a number of other ongoing supervised learning approaches, so classification tasks like classifying lodging or early vigor, or other plant breeder notes that are taken in the field. More detailed networks for doing detection of specific plants. So the detection networks generally find bounding boxes around objects. And this can be helpful for counting plants, but also sort of differentiating between plants and weeds, or other tasks where you want more detailed localization information. And then other approaches for segmentation as well. So to summarize, we're building a number of tools really focused on outdoor imaging, both for aerial and what we would call inexpensive ground-based platforms to extract per plot or per row information. And then developing sort of tailored deep learning architectures to try to accomplish tasks like segmentation or counting or detection. And then at the output, trying to provide some visualization tools that make the data that's extracted from these deep learning architectures more usable for plant scientists. So both visualizing the images, the original images, and then visualizing the resulting trained networks to try to provide a bit more trust and intuition as to what's happening. So with that, I'll just say that, as I said, our group is very, very much open for collaboration. So I'd encourage you to please send me an email, and we can discuss these sort of approaches in more detail. So thanks very much.
Wow, Dr. Stavness, thank you very much for your really exciting seminar in a really cutting-edge area in machine learning and going towards plant breeding there. So we'd like to open it up for the next 15 or so minutes for questions. So you can write those in the chat box, which should be at the bottom of your screen. You can either direct those at myself, Seth Murray, Dr. Stavness, or just address them to everybody, and I can read them then. So the other option is you can unmute and just turn down your volume so we don't get too much feedback, and just ask your question yourself. So I'll give a couple opportunities for that. So while you're thinking about your questions, I always like to make a brief plug for the Plant Phenome Journal. So we're an open access society journal dedicated to advancing issues just like the ones talked about here. And we're also accepting articles that help push the frontiers of plant phenotyping and catalyze this just like the Phenome meeting does for meetings, or North American Plant Phenotyping Network does for societies and organizations. So with that, I'm going to ask one question, and then hopefully we'll get a couple questions from the audience. So this was really cutting edge. I, as a plant breeder, I would envision how I could use a lot of this material, but it seems like a lot of this might be a step away from what the plant breeders are actually, using. So are there other people that sort of interface in between you and the plant breeders, or do you just send them spreadsheets of data, or how does that interaction work?
Yeah, so I think the current interaction is, through exactly that, spreadsheets of data. I think sort of the immediate need from the plant breeders approach is to try to get phenotypic information at a larger scale than is possible from manual phenotyping in the field. So I think that's sort of the initial desire, at least that's been discussed in our group, is to help try to scale up these phenotypic information to make and have it at scale that can be useful for large breeding trials. But then, I think the future direction really do see that there's a lot of additional value other than just the numeric data themselves to provide to plant breeders. So certainly there's a bit of a gap currently, and definitely that we think that there's these sort of magical research associates in between, that have background both in computer science and plant science that can help bridge the gap. But eventually we do see these tools being useful, just as there's, most plant breeders are very comfortable with genomic tools right now. We think that the phenomic tools will get to that point as well. It's just that they're so much more sort of early stage right now that it's it's kind of the Wild West in terms of analysis. But that's why we've tried to focus on these sort of web-based tools that makes it a bit easier. A plant breeder could go to a website, take a look at the pictures, at least together with the numeric data, so that they have maybe a bit more confidence in the measurements that are coming out.
So I guess great answer. Let me just follow up on that just a hair. So you mentioned that there's a social scientist involved, and then also just from a perspective of a plant breeder. I mean, probably 10 years ago, most of the plant breeders were not actually that comfortable trusting in genomics tools. So if there's some challenges, I guess, in interacting between the two cultures and getting some of the breeders to see the value and vice versa for them to sort of convince you of the value of certain things that you may not have realized before?
Yeah, absolutely. I think there's been, as I said, we've been at this for 3 years. There's definitely been an initial sort of feeling out the period where we've sort of calibrated to each other's expectations. But I think that probably the value is still not fully there yet for the plant breeding programs, because they're big operations that they're multi-year studies to produce a variety. So at this moment, we're trying to fit in these sort of analyses into the existing plant breeding. Once I think they see value, then I think you'll see buy-in where maybe some of the processes are adapted to these approaches. But certainly at this point, we're trying to provide or demonstrate that value from these imaging techniques, so that we can move forward in more of a kind of coordinated approach. But I should say, that is one of the strengths, that we have a really strong connection here in Saskatchewan between the plant science groups and the computer science groups, since we're co-located and we're working together. So I think that's absolutely critical, is like the collaboration. So we've been fortunate here.
Yeah, it's really cool. I mean, we've seen these challenges again and again across different people's projects across the country and world. So they're well known, not always well recognized. So yeah. I don't mean to dig at that. I think it's important to present. It's not just an easy thing. So
absolutely.
You have a number of questions. The first one that I see here is from Scott Wild. His question is, can this CNN be used to create simulated plant images, and has that been done?
Yeah, great question. So there's another class of deep learning approaches that I haven't talked about, generally called generative networks, or generative adversarial networks is sort of the most common, or the most popular one right now of this kind of class of approaches. And they're called GANs, for the acronym is GAN, GANs. And those particular techniques are trying to generate images that kind of are representative of a particular training set. So generate new pictures that fit with a particular class of object or style. So they've been quite successful and popularized in general computer vision tasks for generating sort of novel faces of people that don't actually exist, but that look absolutely realistically like faces. In the area of plant phenotyping, there has been some initial work in GAN. So I know that there's a group that's done GANs for those Arabidopsis images. So it's sort of a similar approach that we took to using computer models to try to fill out the training data for Arabidopsis rosettes. They used a GAN model to generate or synthesize these synthetic rosettes. And that was also presented at CVPPP 2017, the group from Edinburgh. So yeah, it's been used a little bit. I think it's still very wide open. They're absolutely powerful techniques, but they have similar challenges to CNNs. Coming up with intuition as to what's happening, trying to actually get the right type of variance you want. Sometimes there's a tendency to produce very similar pictures from those. But yeah, definitely an open area of research.
Great. We got a question from Cynthia Sankaran. She says, thanks for the interesting talk. What are the sample sizes in your studies? What sample size would you recommend for deep learning for decent accuracies? And then did you compare deep learning approaches with other simple machine learning approaches like discriminant analysis?
Yeah.
Okay.
Yeah, that's a good question. So as I said, we're generally restricted in the sample sizes we have based on the amount of labeled data we have. So in the IPPN, the Rosette images, there's a few hundred images in the data set. For the wheat emergence examples, we have I think a small number of large plots. Then in the patches, then we have maybe hundreds of patches that have been labeled. So in general, we're pretty data limited or image sample size limited. As far as how many samples you need, the main thing to check there is if your network is overfitting or not. So a standard approach is to have a training set of images as well as a validation set of images. And while you're training, you check you train on the training set and you're sort of checking performance on this validation set to see when things start to overfit. So the longer you train, the more there's a tendency to do that. And then you hold out another set of images that have never been seen by the network called a test set. And those are what you run the actual analysis on, or the actual accuracy analysis on. So that's a standard approach. I think we're usually limited. So we have to generally adapt the architectures to either make them very small. Another approach that's quite commonly used is to fine-tune a pre-trained network on a small data set. So that's also commonly used where you have a network that's been trained on general computer images of objects, like a Google ImageNet, and then you sort of take that network as it is and then train it a bit more with these plant images, and usually you sort of freeze part of the network so that you don't overfit on those images. So that's another approach that's possible. And then, generally in these approaches, we try to compare to previous approaches whenever possible. So that's the nice thing about having a public data set that's been used by different groups, is that you can look at previous results and try to see a comparison there. But where those don't exist, like in the wheat emergence, and we also did wheat biomass estimations, we did implement sort of previous approaches that would use sort of traditional image analysis or traditional machine learning approaches.
Great. I got one more question that was asked here from Jeremy Edwards. He works on rice, which grows in a flooded field. Do you anticipate any particular analysis challenges or advantages for imaging with water instead of soil in the background?
Yeah, that's a great question. We don't grow a lot of rice in Saskatchewan, so we don't have much experience with that. So I'd be interested to see what those, I assume there are unique challenges to water versus soil. The soil background itself can be quite challenging in outdoor fields, particularly because, in most practical trials, they're done with no-till, so there's usually a lot of crop residue in the background already, so you have that to contend with. But as far as the water as a background, I'm not sure. I'm sure that it is challenging. Then it probably is a good test case for these convolutional neural network approaches for segmentation. But then certainly the thin structures is also generally a challenge. So I think that would be another aspect of rice phenotyping that would be difficult. Great. Oh, I think Seth, you might be muted, maybe.
Yes, I was reading the next question to you on mute. So the next question is from Shay Simpson. A very relevant question, I think, that's probably on a lot of people's minds. Are you developing open source code, or do you see opportunities to protect these techniques? And what is the role, I guess I'll just expand on that, of your university in making these decisions?
Yeah, that's a good question. Yes, we are, where possible, trying to make these techniques open source. So as I mentioned earlier in the slide, we have a platform called Deep Plant Phenomics, where we're trying to take these techniques and sort of catalog them, these sort of reference architectures, making them there and usable. We think that the the field itself of plant phenotyping will advance much more quickly by sharing both code and data where possible. Like certainly with biological data, it can be challenging to make that available, but certainly on the imaging side, you can imagine being able to provide labeled imaging data sets that are kind of a bit separated from the underlying biological question. I think that's probably the way we'll get public data sets actually happening, so that you can train networks to do phenotyping, but not necessarily scoop the person on the biological question that they're trying to answer. And then in terms of the making code available, yeah, we think that increases the impact of our work, certainly. So any of the research papers we publish, we also try to provide code to make that work reproducible, and so that people can kind of extend it themselves. And that's sort of the general standard approach in the area of machine learning. Code and data availability is quite common to make papers reproducible and to increase their impact. I think, particularly in the area of deep learning, I mean, we're generally working on adapting existing techniques into these problems. So I think that the most value is in kind of the know-how that's happening, as opposed to trying to have particular IP around the software itself. So certainly on the research side, we try to make things open source. In terms of kind of adapting some of these tools to plant breeding, those are areas where we're certainly looking for academic partners to try out our tools like plot vision to get per plot segmentations. But there may be also sort of commercial opportunity there with commercial breeding companies to take some of those tools into a commercial space. So it certainly depends on the project, but certainly for the academic projects in deep learning, we try to make source code available. So I do encourage you to check out Deep Plant Phenomics on GitHub.
Great. You've gotten thank yous from most of the people who asked questions. I'm going to open it up verbally for any other questions from the audience. I have one more question, but I'll save that for last. Does anybody have a question? Go ahead and unmute yourself if you'd like to ask it, Dr. Stavness. Okay, hearing none, and seeing no more in the comments, unless you see any, Ian. So under your last slide in visualization tools, your last slide, I'm sorry, on your outdoor section, it looked like there was a barcode actually inside the plot. And I'm just wondering if that's part of the way you're doing non-orthomosaic identification of plots when your GPS isn't all that accurate.
Yeah, so those are.
Oh, I think you're muted, or we lost you.
They're exactly that. Markers in the guards of those fields, and used to do the orthomosaic. So we do a standard workflow to generate an orthomosaic, and so those use those ground control points. So those are useful for doing to transfer segmentations from one flight to the next. So if you've done sort of a semi-automatic segmentation, found all the plot boxes for one date, and you have those ground control points, then the next orthomosaic will be georeferenced, so you can sort of apply the segmentation boxes for the plots onto those subsequent images. Yeah.
So in your slide right now on summary, I mean, I see a ground control point there, and what I was actually talking about was it looked like I don't know if that's canola on the upper right-hand side, but you had a picture with that. It looked like maybe it had a 1-inch square barcode within the plot.
Oh, a little one, yeah. So we've also done some experiments with having small barcodes that we can read, just as kind of a verification. We're extracting plots, and then we use those barcodes as sort of a ground truth, just to make sure that we have the right label for those from that row detection algorithm. Yeah. Okay,
so those are not ground control points then.
If you saw a small little tag in there, those are not ground control points. Those are sort of they're sort of like ad hoc labels that we use to automatically identify plots as a check, basically. Okay. Yeah.
Well, this was great. I don't see any more questions, and I think we're out of time. So let's thank our speaker, Dr. Stavness. You'll just hear me clapping because everybody else is muted, but you had a very large audience, and I will do my best to get this up on YouTube quickly because I know that